Good afternoon, all. My topic is acetone butanol fermentation. Looking into the history of acetone butanol fermentation, in 1862, Pasteur discovered butanol fermentation in bacteria. In 1911, Fernbach described several species of bacteria which conduct acetone butanol fermentation. In 1916, acetone butanol fermentation was developed by Cham Weizmann using Clostridium acetobutylicum from fermented green products such as corn. Microbial fermentation is used for the production of acetone butanol in Europe and America until 1970s. Currently, the microbial fermentation is used only in China, and the microbial fermentation was replaced by petroleum or chemical based industries. Butanol is used in automobile industry for the production of latex, enamels and nitrocellulose, and butanol is also used as biofuel. Acetone was used as explosives and used for the production of synthetic rubber. Acetone was used as explosive mainly during the time of World War I. Microbial fermentation. The raw materials used for acetone butanol fermentation are starch, molasses, hydrolyzed wood, sucrose. Molasses are the byproduct of sugar industry. Products obtained after acetone butanol fermentation includes butanol, acetone, butyric acid, acetic acid, isopropanol, hydrogen, and carbon dioxide. Mainly, three major types of fermentation are carried out by the Clostridium species: acetone butanol fermentation, butanol isopropanol fermentation, and third, butyric acid acetic acid fermentation. Acetone butanol fermentation carried out by Clostridium acetobutylicum using corn media. Next, Clostridium secara acetobutylicum using molasses media. Butanol isopropanol fermentation carried by Clostridium butylicum. Butyric acid acetic acid fermentation carried out by Clostridium butyricum. Clostridium beijeringi has also been used with good results. Clostridium species are anaerobic, motile, spore-forming bacteria. Acetone-producing bacteria first produce acetic acid and butyric acid and then convert to acetone or butanol. That is, first acid is generated and it is converted to solvents. Clostridium cultures can be maintained as soil stocks as long as 30 years. The sluggish growth during fermentation and poor conversion of acid for the corresponding solvents indicate the degeneration of cultures. Such cultures can be rejuvenated by heat shock treatments. Heat shock treatment is done as follows. Spores are suspended in appropriate nutrient media and they are subjected to boiling about 90 seconds and rapidly cooled. Vegetative cells and heat resistant spores are selected by this process. The remaining spores are incubated till still sporulation. Again, heat shock treatment is applied and the process repeated till selection of the most heat resistant spores. The acetone butanol fermentation is biphasic, which includes acidogenesis and solventogenesis. In acidogenesis, acetate, butyrate, hydrogen, and carbon dioxide are formed. In solventogenesis phase, butanol, acetone, and ethanol are formed. The fermentation includes mainly three steps. First step is production of spores. Spores are added to tubes of nutrient media to develop inoculum. Potato or corn medium is used in case of corn cultures and the organism used is Clostridium acetobutylicum. Potato or glucose medium used in cases of molasses culture and the organism used is Clostridium secara acetobutylicum. The spores are added to nutrient media, heat shock treatment is done, and after rapid cooling, incubation is done. Clostridium acetobutylicum is incubated at 37 degrees Celsius, and Clostridium secara acetobutylicum is incubated at 31 to 32 degrees Celsius for 20 to 24 hours. After production of spores, the next step is preparation of inoculum. Inoculum developed by the above mentioned method under strict anaerobic condition is used for inoculating fermentation media. Heat shock followed by rapid cooling ensures anaerobic conditions, which is later maintained by evolution of carbon dioxide during growth of microorganisms and incubation under anaerobic conditions. The building of inoculum is done in stages, starting from smaller volumes to larger volumes to ensure great yield and resistance to contamination. The prepared inoculum is added at 0.03 percentage volume per volume, and low inoculum ratio about 1:3000 is used. The next step is the fermentation. The medium used can be corn or molasses media with corn steep liquor, CaCO3, ammonium sulfate or ammonium phosphate. Inoculum added at 0.03 percentage volume by volume. The corn media have a pH of 6 to 6.5 and molasses media having a pH of 5.5 to 6.5. Incubation is done at 36 degrees Celsius for 36 hours. Fermentation is carried under strict anaerobic conditions in huge fermenters about 5,000 gallon capacity. The fermentation occurs in three phases. First 18 hours, 0 to 18 hours, rapid growth occurs. Acetic acid and butyric acid are produced. Evolution of CO2 and pH drops to 5.2. During the next 18 hours, acids are converted to acetone and butanol. There is increase in pH value, and also there is sharp decrease in titrable acidity. As the pH as acidity decreases, and this condition is called acid break, and products are ready for harvesting. The acid break indicate the products are ready for harvesting. And during the last 18 hours, growth and solvent production is stopped, and there is only low rate of gas evolution. The products should be recovered before this phase. This is the figure of acetone butanol fermentation. First, glucose is converted to pyruvate by glycolysis, and from pyruvate, acetyl-CoA is formed. As we said earlier, there is acid forming phase and solvent forming phase. In acid fermentation phase, acetate and butyrate are formed. Acetate is formed from acetyl-CoA and butyrate is formed from butyryl-CoA. In solvent fermentation phase, three products are formed: ethanol, acetone, and butanol. Ethanol is formed by the reduction of acetaldehyde. Acetone is formed from acetoacetate, and butanol is formed from the reduction of butyldehyde. Sometimes ethanol is also produced by the reduction of acetaldehyde during the fermentation. In corn media, the ratio of butanol, acetone, and ethanol is 6:3:1. In molasses media, the ratio of butanol, acetone, and ethanol formed is 6.5:3:5. Along with the butanol, acetone, and ethanol, carbon dioxide and hydrogen are formed. Carbon dioxide, which forms about 50%, and carbon dioxide about 2%. Autolysis of cells in the last stages of the fermentation result in the release of riboflavin. Last step is product recovery. Acetone, butanol, and ethanol is recovered through continuous distillation and fractionation. Carbon dioxide after fermentation is converted to solid carbon dioxide, solid CO2, and is used as dry ice. Residues left after distillation, including biomass and unspent medium, are dried and used as animal feed, manure, etc. Thank you.